All right, good afternoon. We do have a little bit to talk about to finish up Chapter 8, and so I want to talk about that topic right now. And then we will be done with nucleic acids. Move on to lipids next week. So, when you look at the DNA as a chemical, its purpose is to archive information. And so, even though it's very stable, you've got to think about the ways in which it is most unstable. The places where even a small amount of reaction will happen that will change its structure. Because if a reaction changes its structure, it changes the information, and the archive is no longer valid. It has an error in it. And so there are bad chemistry that can happen to your DNA, and all of these things you could actually sort of predict what's going to happen to it, what could happen to it based on your organic chemistry knowledge. Now they are moved in order, they're arranged in order from small to large changes. And the first one we'll talk about is deamination. And sort of small, sort of specific to general. So deamination is small and specific change that can happen to your DNA. We've already talked about how things can change to the enol form. And if you look at it, if you remember your organic chemistry, you probably had some reactions in which the water would react with a amide that is just sticking out there off the molecule. Over time, the N can be replaced with an O. And in fact, that's exactly what can happen. And if that happens to an N-containing amino acid, an amino acid with a primary amide sticking off of it, then it will change it from a hydrogen bond donor to a hydrogen bond acceptor. This means it will change it entirely. It will change its base's name. And so if you look at cytosine, cytosine has an NH2. If that changes to a C double bond O, it becomes uracil. Completely different signal being sent because you've changed the hydrogen bonding pattern. Now if you have methylcytosine, that gets deaminated to thymine. But look at this, there's some reasonability to this. Because cytosine can turn to uracil naturally, in DNA, you don't have uracil naturally. You have it in RNA, but RNA is not supposed to archive for a long time. In our archival system, we actually have a failsafe in for if the cytosine gets deaminated and it becomes a uracil, what we can do is we can make a DNA binding enzyme that will look for uracils in DNA. And if you find a uracil in a DNA, that DNA means it's not supposed to be there. And so it must be a deaminated cytosine, so you should fix it. And in fact, you have exactly this kind of uracil binding and uracil modifying. It like fixes the uracil. So this might be the reason why we use T instead of U, because it's a methylated form of uracil. And so we can know that it's right if it's in the DNA. Adenine can be changed, and guanine. Each of these three nucleotides can be changed from one thing to another, but only cytosine becomes something familiar when it's deaminated. So this will also happen with amino acids. If you think about it, the asparagine has a susceptible nitrogen, and it can actually over time become asparagine to aspartate. I actually saw this in the lab. I had an old peptide that we did mass spec on, and we found out that the unit was off by 1. And that's because it had been deaminated over time. So this will happen even in the tubes. You got to be careful if you're working with old peptides or old proteins. And then finally, there actually are chemicals that promote deamination. Nitrous acid in particular will promote it. And it will actually, I used it in high school for an independent research project. And I was not as careful as I should have been because I didn't have safety oversight. I didn't have a lab safety training video. And so after I didn't use gloves, and my fingers actually started to turn yellow. And I said to myself, oh, maybe I should wear gloves after this. So wear your gloves when you're working with nitrous acid. Nitrous acid is actually a carcinogen. It's not that bad as an acid. I mean, it's pretty bad as an acid, just as an acid. But because it can do this to your DNA, it can actually produce tumors from this deamination mechanism. So moving one step up, if you can remove an amine or change an amine, you can actually break one of the other susceptible bonds. And if you look at the bonds, you have that It's basically a hemiacetal bond, or no, it's a full acetal bond that you have right there that's formed to the purine. And it turns out that these bonds are susceptible to hydrolysis as well. In fact, for some molecular orbital type reason, guanosine is particularly susceptible. Its bond is can be attacked by water, and then the guanine can be cleaved off, leaving an apurinic residue. You know that there's a pyrimidine on the other side, so it needs to be that wide to fill in the gap. And for some reason, this doesn't really happen with pyrimidines, and it happens more with guanosine. But the main thing is, if you look at an old strand of DNA, some of the guanosines will actually be gaps. They'll be like missing a tooth. But the thing is, you can also make an enzyme that looks for that. If you have the guanine missing for a while, then you can actually have some weird chemistry going on. For example, if you have an abasic site, then it can be oxidized to form an aldehyde, and then that can react with the thing across from it. So here's an example where they saw this happening. The apurinic site, guanosine, has been knocked out by hydrolysis, and if you end up with an aldehyde group on there, that can form a covalent bond, and it can actually attack to an adjacent adenine, like in the next strand over and the next base down. And once that happens, that's a very hard thing to fix. It's pretty easy, and you have mechanisms to fix the missing residue, but when you start to have covalent bonds happening, then you start to get that entire gene, that entire DNA helix is messed up. So moving on up, we actually have an interaction between two adjacent residues. This happens most often, for some reason, between two adjacent thymines. Remember that UV light can be absorbed by the bases, but that means also sometimes the UV light puts extra energy into them and can move the electrons around and can create covalent bonds. So here's a case where you have two adjacent thymines and you have exposure to UV light, for example, in the skin. You will often have a tumor that can result from that because you have a biochemical reason for it. You have a covalent bond being formed between adjacent thymines. And if you just look at this, yeah, these random covalent bonds are bad news, and they can happen wherever you have a TT sequence. And so sunlight can do this, the UV in sunlight, cosmic rays and radioactive decay emissions. Basically, if it can be absorbed and put energy into the thymine dimer, it can rearrange the electrons, and then it's very hard to fix. This is a model. I went to a museum that made a DNA model out of Legos, which I really liked, and these have magnets between them so you can peel them apart and stuff like that. It's really cool. But the one thing that they showed is they showed a cyclobutyl TT dimer, which is what happens with this. This is on the left, they have the actual structure of it, but on the right you have the model of it, and they showed that if you link those two T's in a bad way, you introduce a kink into the backbone, and that kink is shown by the broken backbone in the model. So now moving on up from that is more, those are the specific changes that can happen. You can have more non-specific changes you can happen if you have a reactive chemical that forms a covalent bond somewhere with your DNA. You have an alkyl bond, you have alkylation that can happen. And you can have deamination that results from this. Basically, alkylating agents will add something onto DNA. In the simplest case, it's a methyl group. And there's even this cofactor, this S-adenosyl methionine. We haven't talked about it this quarter yet, but its job is to add methyl groups onto things in the body. You'll talk about it in biochem too. So that's a reactive methyl group, and if you look at that, you don't need to know what it is to see, oh, that's a reactive methyl group in the middle there. If this cofactor gets next to your DNA, it will actually alkylate it and put random methyl groups onto it, and that can change its message and cause cancer. Then you have alkylating agents like these things with these obviously reactive methyl groups in the first two cases. In the bottom case, you have a reactive leaving group that will leave this long carbon chain covalently bound when it leaves the molecule. But notice the name of this one at the bottom, nitrogen mustard gas. I thought I had it highlighted, but there you can just look at it. Look down at the bottom, nitrogen mustard, and it's the major component in mustard gas, which was one of the first poison gas chemical warfare agents. developed in World War I. And it's sort of depicted in the first Wonder Woman movie. The villain is kind of like is a German scientist who works on poison gas, and that really happened. And it happened because it was bad news, because it was an alkylating agent that would mess up everything. It'll definitely mess up DNA if it's able to form covalent bonds. You don't really have a rescue mechanism, an enzyme that's going to fix that too easily. So nitrous acid can also act as one of these agents, and because it can form nitrosamines, and those are really reactive. Basically, if something's really reactive, if you look at the structure and you say to yourself from your organic chemistry intuition, oh, that looks like it's a weird structure, then if it's a small molecule that you can have in great quantity next to your DNA, you want to get out of there. You want to get it away from your DNA because it's going to mess up your DNA, and everything else in your body can be replaced. How do you replace it? You go and look at the DNA. So if it starts to mess up your DNA, it's messing up the permanent instruction book for your cell. And that's why you get things like cancer happening. Here's an alkylation thing that can happen to guanine. It happens through the guanine tautomer. The guanine tautomer will form an OH that is susceptible to methylation. And that will clearly change, that will lock in a different hydrogen bonding pattern, which means when that DNA strand gets replicated, you'll have a different base on the other side. Here's another methylating reagents can also do other things. Here's an example of a 2-methyl guanosine. If a methyl gets in the wrong place on a guanosine, then actually it can react. That makes a reactive guanosine that can form a covalent bond crosslink to lysines in the histone proteins. You know that lysines can form covalent bonds under certain conditions, and there's lots of lysines in histones. So the lysine can actually covalently react with methylated guanosine. And it looks like this happens, this messes up your DNA too. So remember that whole story about methanol, using that, when you drink methanol you create a lot of formaldehyde, and that makes crosslinkers, and that's why methanol is poisonous. Remember that ethanol is what you use for that, and ethanol actually also makes an aldehyde. It's just that this aldehyde isn't quite as reactive as formaldehyde, but it's still reactive enough to mess up your DNA. So chronic ethanol exposure can create the acetaldehyde, which can do this to your DNA. And if you look at this, just look at the red marks. You have your DNA being really messed up by covalent bonds to carbon, because you have created this reactive aldehyde that happens when the ethanol is converted to an aldehyde by the liver enzyme that's trying to get rid of it. So this is one of the ways in which ethanol can mess up your body. Not as reactive as methanol, but it can still mess up your DNA because your DNA is very sensitive to this kind of thing. And finally, if you think about covalent bonds, if you remember about free oxygen radicals, I mean, we need oxygen to live because it's reactive, but also because it's reactive, it creates electron-rich radicals that have reactive electrons on them. And they're so reactive, they basically can go anywhere and they can break pretty much anything. So you want to keep them away from your nucleus. The main thing that produces oxygen radicals is oxygen-changing chemistry. The mitochondria use oxygen, and so they produce a lot of radicals. So you can see right now why it might make sense to have separate rooms for separate chemistries. You have a room for the oxygen chemistry, which can produce oxygen radicals in the mitochondria. And you keep your DNA as far away from that room as possible. You put it in the nucleus, and you have 2 barriers between the 2 rooms. So that's an oxidative damage. There can be so many oxidative radicals around that they can actually cause double-strand breaks, and those are really hard to break, to repair. Because if you have a single strand broken, well, then you can still use the other strand as a template, right? You can still copy it. But if you have both strands broken and they fly away from each other, well, then the DNA can only be half copied, and then you have a more severe problem that's harder to repair. But I've already mentioned that DNA actually there is some saving grace in the form of enzymes. There are enzymes that have evolved to repair these problems. For example, to find uracils in your DNA, and even to repair some double-stranded breaks. And so that's chapter 29. That's a long way away, but that's at the end of Biochem 2. And but at the same time, oxidative damage, you don't want to do any more than you have to. And one of the things that you can do is you can consume antioxidants. There are some studies that purport to say that consuming green tea, which has antioxidants in it, can actually specifically prevent DNA damage. Now, these studies are not the strongest studies, but I still think it's a good idea. And by the way, I like green tea. So, it's a win-win for me. And in fact, those alcohol-derived crosslinks that we showed you, the reason why I showed you that a second ago is because even that can actually be fixed, because you can have you can have double strand you can have fixing of even stuff that big through double strand swapping, like recombination, which is another Biochem 2 topic. Really cool chemistry, but we'll have to wait to learn more to see how it's fixed. And so the other thing is there are different things that use this chemistry, and I have a couple of examples of DNA damaging chemistry that we can actually use. Because oxidative radicals damage the DNA, and iron produces oxidative radicals, especially in its iron 2 form. It's actually one of the reasons why iron 2 is kind of dangerous and why we want, even though it's very useful chemically, it's a little too useful. It creates these oxygen radicals, and then those can create breaks and those can damage your DNA. But of course, sometimes we want to damage the DNA, like we want to damage the DNA in a tumor. What if we had a DNA targeting thing that would bring these iron-generated oxygen radicals close to a DNA strand that we wanted to mess with? So here's the first idea. If you have just a flat conjugated molecule, it can actually sit into between the rungs of the DNA ladder. It's called an intercalator. Okay, and there's a foundations problem about ethidium bromide, which is an intercalator. And so this is an example of where you use a molecule like that, and then you attach an iron group to it, an iron 2 atom, that will create a lot of oxygen radicals. So this will go to a specific place, this will go to a place between two rungs in the DNA, and this will produce a bunch of oxygen radicals, which will break the strands. And you can actually see this working in the lab. So that's not too specific. That's not a very specific agent. But the interesting thing is, if we take this same idea and we make a molecule that is more specific to a certain strand of DNA, then we can have something that targets a specific part of DNA, and we can have the iron effectively breaking the DNA at a certain point. The way that works is this is a form of iron that is bound to, you see it has all these nitrogen-containing imidazole type rings. And so if you make this, it actually will bind in the minor groove. And it will fit into the minor groove depending on how you alter it. You can make it fit into a specific place in the minor groove. This is more proof of concept, just that it can fit into the minor groove, but it fits at this particular place, and it places the iron molecule in a particular location at the DNA. That means that the iron is producing all the oxygen radicals, but it's producing it in one place on the DNA. And that's producing a lot of breaks in the strand. And what it will do is, you see how you have the DNA sequence at the bottom there. This will produce a lot of breaks in the 5' strand on the top and in the 3' strand on the bottom in this particular location, because those are the places where the backbone is close to the iron. It's all based on the structure. And so what will happen is, you can run a gel like this. You can throw this thing at a piece of DNA, you run a gel, and then you actually separate you basically label the 5' strand so you see its pieces, or you label the 3' strand so you see its pieces, and you see that you have this sort of complementary fragments that are happening. The fragments that are happening exactly where the iron is. And so you can see this is doing what you want it to do. So at this point you have a specific DNA breaking piece of iron. The only thing you need to do is work on the chemistry of the molecule to see if you can make chemistry that binds a specific sequence of DNA in the minor groove. Then you will have a DNA cleaving chemical. So that's how you use gels to measure how the DNA cleaving chemicals work. And there are similar gels that were used in the polymerase context. So you remember we use those for DNA sequencing and things like that. If you have a polymerase, it's going to make a lot of DNA, and you can do things, you can measure where the fragments are. Well, what if instead of making the fragments from having deoxynucleotides in there, what if we had the fragments that are made by having something that is messing with DNA transcription and having it like fall off the DNA, so that the fragment happens when the polymerase falls off the DNA or is messed up somehow? And that's exactly how remdesivir was tested. So I just want to show you this. This is the last thing that we have to say. But it's a direct biochemical gel that you run to show that remdesivir works on the viral polymerase. It's an RNA-dependent RNA polymerase. It's how the virus makes more virus. The virus makes more RNA genomes by using an RNA polymerase. And so it's very important to the virus. Remdesivir actually interrupts this polymerase, and that's how remdesivir works. So if you look at how the polymerase works, you see 1, 2, 3, 4 on the right side of this screen. And you can see that if you have RNA synthesis going on, you have a growing RNA chain being produced from a template by the polymerase. If you have remdesivir around, then remdesivir will replace ATP, and it looks enough like a nucleotide, it can actually sit in the ATP. And if you look at remdesivir's structure, it actually has a 3' OH. So the chain can continue to grow. But what you see with remdesivir, it's a little bit like a time bomb. The remdesivir sits in there for a little while, and the chain grows for another 2 or 3 residues, but then it falls off, and the remdesivir does something that makes the chain break after 3 or 4 residues after it is added into the growing chain. So it's important to understand remdesivir works by mimicking an adenine residue, an adenine base, that it actually goes into it, but then something happens, and it goes on for a little while, but then it dies. And here's the structure of exactly what's going on. Remdesivir is shown, I believe, in green over here. If you look over there, that you see the green molecule, it's the remdesivir. And you see that there are another 3 or 4 yellow molecules that are added on after the remdesivir is incorporated into the chain. And the way that shows up is we know that's true because we see it on a gel. We can see where the remdesivir is added, and we can see that there are always 3 or 4 more nucleotides that are added after the remdesivir is incorporated. And so they do this in the context of radiolabeled phosphate. That's the way they can see these gels. And it's why we don't do these gels, because I don't want to mess with radiolabeled phosphate. And I'm not going to have you interpret this data specifically, but I want you to see the top of it and see how the top looks like something that we've been dealing with. It looks like a polymerase. It looks like a PCR type reaction. And you can see the same kind of elongation polymerization reaction is going on, and remdesivir interrupts it. You can actually go through and you can see how remdesivir appears to work slightly differently. You see down at part B, they have lines for MERS-CoV, SARS-CoV, and SARS-CoV-2. So these related coronaviruses also bind remdesivir. And we're not going to get into interpreting the data from this, but mostly I want you to see, if you look at the bottom there, look at the G is where you have the chain termination, and then you see that you have I plus 1, I plus 2, I plus 3. That shows you if you have remdesivir added in, then you will actually have 1, 2, 3 more things added on, and sometimes the chain can keep going. So it's a little bit complicated, but the thing I want you to see is I want you to see how a gel is used to see how far along the polymerase gets after the remdesivir. And you can really get into it. I've assigned this other years when we actually looked at the biochem data. The last thing I want you to see about this is an enzyme reaction. So guess what? They report their findings with a Vmax, a Km, and a Vmax divided by Km measure that they use to talk about the overall efficiency of the enzyme. This uses enzyme kinetics terms. This is biochemistry. So you can do biochemistry enzyme kinetics with gels, and you'll have some simple homework problems that are like that. They're not going to be as complex as this, but I want to show you that it has a real-life reflection in a really important paper showing how remdesivir works. Okay, so here's the homework assignment again, in case you want it again, but of course it's also on Achieve. And have a happy weekend. I will see you on Monday to start the lipids chapter. All right, see you.